Cool. Okay, we'll stop mumbling up here and officially start the event. Thank you guys for coming out on this Wednesday evening to our final lecture as part of the Science in the News lecture series this spring. It's hard to believe the semester is almost over. How many of you have been to a lecture before? Cool. How many of you have been to all of the lectures this season? One? All right, that's okay. That's a high barrier. Try again next semester. It's okay. How many of you is this your first Science in the News lecture? All right, okay. So some new faces, great. Well, we have some flyers up here if you want to see what you missed out on earlier in the semester. The good thing for both this semester and many, many years past, this is our 19th year Science in the News being around, we have all of our, not all the way back 19 years, but we have many years worth of lectures archived. So you can search our website, and if you see something cool, you can watch it there. And yeah, so most of you have been here before, you've heard the spiel, but I'll go ahead and say it one more time. So this lecture series is put on by Science in the News. I'm Dana, I'm one of the co-directors.
I'm Allie, the other co-director.
Yeah, and Science in the News does a whole bunch of stuff under kind of the umbrella goal of trying to communicate cool science to non-scientists, and to do that in a fun and engaging way, and to help teach graduate students such as ourselves, we're all graduate students, how to better communicate that science. So we have these lecture series where we have graduate students kind of work to help the speaker, who's also a graduate student, refine their communication skills, make their slides really great, so that we can give hopefully a high quality lecture to you guys. We also have a really great blog. Actually, one of our editors-in-chief is right here. And they post multiple times a week, really cool articles, long articles, short articles about science stuff that's in the news, as well as things that our members are kind of experts in or find interesting. And there's a ton of great content on our website. So when you're there to check out the past lectures, you can read some of our cool blog content. We also have a bunch of other events. I'll just go through some of them quickly. We have a science cafe called Science by the Pint, where we bring kind of scientists from the Boston area and all of their graduate students and postdocs to a lab and kind of almost do more of a two-way conversation, speed dating kind of science talk if you're into that, or if you want some beer with your science. And I'll let you talk about it.
Yes, and we have a really fun event coming up on June 2nd called DayCon, which is our day-long scientific conference. So if any of you guys are interested in learning about what future technologies we'll be experiencing in the coming years, like CRISPR, genome editing, engineering the weather to try to manipulate sort of climate change, things like that, artificial intelligence, you guys should definitely check it out, and you can go to our website and register for it today.
It's free. You even get a free lunch. And yeah, you'll get to hear cool talks. There's
an interactive session with different museums. Can be really fun.
Yeah, so if you're at all interested in that, we highly recommend it. We moved to a bigger venue this year, so we won't have to, hopefully, knock on wood, cap registration, but please do register. It'll be a lot of fun. Cool, so yeah, well, I guess you want to explain how tonight's event will go?
Our lovely speaker, Shirley, will be giving a presentation entitled Much Ado About Mumps, and her talk will be in a few different parts, so we ask you to hold your questions until her question breaks. We'll have a little intermission about halfway through. And at the very end, we ask you to fill out our audience survey so we can take this data to help our speakers and also help us improve the lecture series. So,
yeah. All right. There we go.
Take it away, Shirley.
All right.
Hi, everyone. As Dan and Allie just said, my name is Shirley. I am a graduate student. For one more week, I am a graduate student. Here at Harvard, and I study virus outbreaks, and I'm here to tell you a little bit about one of the outbreaks a little bit closer to home, mumps. So we'll talk a little bit to start just about mumps and why it's such a big deal and why everyone's worried about it. And then we're going to get into the methods that I use in my lab to actually study mumps and other virus outbreaks. And that's using this idea of genomics to study transmission of viruses. And then we're going to get into some of the questions that we can answer with genomics, like how the vaccine plays into this whole outbreak. So I just wanted to start off talking about this idea of big, scary virus outbreaks. And these are a few virus outbreaks that you might have heard about in the news over the last few years. There was an Ebola outbreak in 2014 to 2016. And more recently, you might have heard about a big Zika virus outbreak in the Americas. And often these sort of spur these crazy news cycle. There's lots of information, and then they sort of go away. And what I think is interesting is that a lot of times when you hear about Ebola and Zika, it sort of feels like the outbreak is happening over there. Like it's not going to affect me. You don't really think about like the implications for how you might think about if you might get infected. So today I'm actually going to tell you about an outbreak that's happened much closer to home and how we think about that more locally. So I'm going to talk about mumps virus, as you probably can tell from the title of the talk. And mumps is an interesting outbreak because not only did it affect the United States, but it actually had a lot of cases of mumps right here in Massachusetts. Mumps is relatively common in the rest of the world, but hasn't been in large numbers in many years. And we'll get into that a little bit later. And not only was it sort of a big deal here in Massachusetts, but actually a lot of the cases were right here on this campus. Hopefully not in this room. So you might have remember a lot of press about Harvard students getting mumps. And more generally, there were lots of cases at other universities in the Boston area. So what I'm going to do is sort of talk to you about all of the questions that arise when you think about a mumps outbreak right here. So I think it caused a lot of thought, like, why did mumps here to begin with? You know, I thought there wasn't mumps in the United States. How did it get here? Am I going to get mumps? But I'm vaccinated for mumps. Am I still going to get mumps? And so we're going to go through all of those questions and talk about how we're using genomics to answer them. But before that, I just want to talk a little bit about what is mumps. So mumps is distinctly characterized by swelling of the jaw. It's a symptom called keratitis. And anytime you see like mumps infographics, this is my favorite little infographic from the CDC, you see these swollen cheeks, but you also have lots of other flu-like symptoms, fever, headaches, things like that. And mumps is caused by a virus, and it's transmitted primarily by saliva. So typically you see big mumps outbreaks in groups of people who live really close together, like college students. So often sharing of saliva happens when people are sharing drinks or they're living in really close quarters. So that's why we see these sorts of outbreaks. And I'm also going to talk a little bit more about the mumps virus itself a little bit later in the talk. But first I just want to talk about the history of mumps outbreaks and why it's actually so surprising that we had mumps here in Massachusetts. So mumps used to be sort of a ubiquitous childhood disease. Did anybody in the room have mumps actually? Yeah, people think well. And often people, like adults in certain age range, have had mumps because the mumps vaccine was developed in the late 1960s. And so before that, most children in the US and elsewhere had mumps. And there were nearly 200,000 cases of mumps every year in the United States. And although the symptoms that I described, fever, headache, don't sound that bad, mumps can have a lot of serious complications. So it can cause infertility in men, it can cause deafness, and it used to be the leading cause for hospitalization for meningitis and encephalitis. So as a result, it was one of the viruses that was a candidate to develop a vaccine. The vaccine was developed in the late 60s, and you can see really clearly from the graph I'm showing here that the number of cases of mumps dropped drastically after the vaccine was introduced and continued to drop even more when they started recommending a second dose of the vaccine. And that's because they grouped the mumps vaccine with measles and rubella. So MMR, and kids have had the MMR vaccine, that's sort of where it comes from. And so after people started getting multiple doses of the mumps or the MMR vaccine, the incidence dropped to almost no cases. In 2003, there were fewer than 200 cases in the entire country in that year. So we really saw a stark decrease in the number of cases. But if we look at what's happened a little bit more recently in mumps, we actually see that there's been a drastic increase starting in around 2006. So in 2006, there was a large outbreak, again, on college campuses in the Midwest of the United States. And there were over 5,000 cases there. And then we saw a little bit of a drop, but still more mumps than usual in the intervening years. and then a much larger outbreak in 2016 and 2017 that affected not only Massachusetts, but all over the country. And there were over 11,000 cases reported. So this is a lot bigger than just the 200 we were seeing in the early 2000s. And so that's a big reason why we want to study this outbreak. Why is it coming back? Why are there so many cases? And I'm going to start talking about one of the sort of tried and true methods for controlling outbreaks, not just mumps, but really any virus. And that's this idea of contact tracing. And this principle is actually quite simple, but it's been used to successfully contain all sorts of outbreaks all around the world for decades. And the idea is just, if one person is infected and they come into the clinic or the hospital, you ask them, who did you come in contact with recently? And depending on how the virus is transmitted, whether it's by saliva, by blood, that might narrow down the people a little bit. But essentially, you try to identify all of the people they might have contacted, and then you go follow up with all those people and determine if they were infected and if they had any other contacts. And that's one way of really tracing exactly how a virus is spreading, and also trying to contain it by trying to prevent people from contacting people that are likely to infect, for example. But this method gets a little bit complicated for a few different reasons. One, the outbreak gets really big. Suddenly the number of people who are infected or contacts is growing and growing and growing. It's really hard to track all of those people down, figure out exactly who's infected, who infected who, things like that. It also gets tricky when somebody might not know exactly who they got sick from. With a disease like mumps, if you're at a big dinner party and you might have shared food with any number of people and that was 2 weeks ago, not everybody remembers exactly who was there and also who they might have gotten sick with. And mumps also takes, usually a week or two before you develop symptoms. So trying to figure out exactly who had symptoms in the window, it just becomes really difficult. And another thing, which is like a little bit of a tangent for this talk, but I'll just mention it because I think it's interesting, is the idea that mumps is just transmitted between humans, but sometimes there are viruses that are transmitted in other ways. So for example, I mentioned Zika on the first slide, and Zika is also transmitted by mosquitoes. So if you get bit by a mosquito, you can get Zika. And so now you have this whole other layer that's going to make figuring out who infected you a lot harder. It could have been a mosquito, and who knows where that mosquito is today. So what I'm going to tell you about today is how we use genomics to supplement and augment what we can learn from contact tracing and really try to answer some of these questions about how did the virus get here? Am I going to get mumps? Does the vaccine work? So let's just start with a really basic review of what is genomics in the first place. So genomics, very broadly, is the study of genetic material in organisms. So you probably know that inside all of us and inside any organism is a bunch of cells, and inside each of those cells is genetic material, DNA in our case. And all organisms have DNA, genetic material, including viruses. And so when you are infected with a virus, there's actually both your DNA and the virus's DNA to think about, and I'll talk about that on the next slide. But first, I just want to mention that you might have seen DNA represented as a series of letters. And so basically, each of these little lines on the DNA represents a molecule that we've labeled as A, T, G, or C. And using a technique called sequencing, we can basically identify what is the sequence of all the molecules in your DNA or in the virus's DNA, and use that to basically get a readout that we can analyze and we can look for patterns in. And so what I'm going to tell you about is exactly this, using these sequences of DNA, in this case from the virus, not from the human, so using the sequence of the DNA of the virus to understand what's going on in an outbreak. And in order to do this, we first have to get the sequence of the virus sample. And the way we do this is we usually start with a fluid sample, a fluid in an infected human's body that might contain the virus. So sometimes that's blood, sometimes that's urine, and in the case of mumps, that's often saliva. So they'll swab your cheek if you have mumps, and that's because mumps is an upper respiratory virus that often lives in your saliva. And what we do is we can use methods in the lab to extract the DNA out of the liquid. And when you do that, you get a combination of the human DNA and the virus DNA, because you're just sort of pulling out any DNA that exists in that sample. And again, in the lab, we have methods that basically wash away the human DNA, and we're left with just the virus DNA. And this is what we, in very tiny tubes, put on these fancy sequencing machines, which is really like large, incredible pieces of chemistry that we use to get this sequence out, that's telling us exactly the sequence of molecules. And because every organism and every virus has a slightly different sequence of molecules, it's the patterns in here that's really going to tell us what's going on. So there are a few principles we have to think about when looking for these patterns. And one is, where do the differences come from in these sequences of DNA? So I think it's very easy to understand that I have very different DNA than a virus because we look really different. But there's also variation in the DNA within individuals of the same species. So I have brown hair and Allie has blonde hair. Maybe that's a difference in our DNA that causes that. And same thing with viruses. They sometimes have very small changes that can have effects on the virus, and sometimes they don't have effects. And that's because mutations happen randomly over time. So every time an organism or a virus replicates its DNA, sometimes mutations happen, and there are small changes to the sequence of letters. And that happens with any organism, basically every time it reproduces. But the reason this is so interesting with viruses is because viruses replicate really quickly. So while it takes a human, 20, 30 years to reproduce, a virus does that in a matter of hours. And so you can see these changes happen really quickly, even in the course of an outbreak. So in a 2-week period, a virus is going to replicate its DNA enough times that you actually will accumulate lots of different changes, and you'll start to see patterns. And so for each person that you're studying, for example, in a virus outbreak, if you take their virus DNA out of there, they might look slightly different. So here, for example, you might take samples from additional people, and they might have slightly different variants that have happened sort of randomly over time. But sometimes these variants are helpful for the virus. Maybe they make the virus easier to transmit or easier to get inside your body. And then we see that those mutations stick around because all of the viruses that survive, that are replicated, are the ones that have these mutations. And so this is very much the idea of natural selection. If you remember Darwin's finches and why we sort of see patterns in what we see over the course of evolution. But again, we can see it in basically real time in viruses. So for example, if this random A mutation happens to help the virus, you might see it in all the other patients who get infected afterwards. And now you can see a pattern, right? Now you know that maybe all of these people, person 5 through 9, got infected after person 4. And so you can start to think about how that might help you track what's going on with the spread of the virus. So I'm going to talk about a really specific way to analyze this kind of data and the subset of biology that's called phylogeny. And phylogeny is basically the study of evolutionary history. And so I'm showing here very small part of the tree of life, which is something you might be familiar with, where you show this tree-like structure. It's very much like a family tree, if you're familiar with that, to show how related different animals are to each other. So I don't exactly know what animals are on here, but you might see that 2 apes are more to each other than they are to a dog, or something like that. And again, this tree happens over the course of millions of years, but the tree that we're going to build from the viruses can happen in the course of weeks because of how fast that they reproduce. So now let's go through a really specific example of how you might construct this sort of complicated looking tree from virus samples. So let's say we have a person, they're infected with the virus, and we're able to sequence the virus and get the sequence out. So this is just a fragment of that sequence shown here. And that person might infect 2 other people in the course of the outbreak. And in the course of... Okay, not sure. Point counter problem. one moment, please. Never happened.
Uh-huh.
Okay. Sorry about this. I guess my computer really doesn't want me to talk about biology.
That's right, yeah.
All right, we're back. Maybe my computer just got infected by a different kind of virus. All right, so back to human infecting viruses. This person might have infected 2 other people. And when the virus replicated during this process, a couple of mutations might have shown up. And those are indicated with the black here. And they're different because they're just sort of happening randomly over time. And then maybe the person on the left infected 2 other people. And they each have a mutation, but they also still have this gene mutation that this person had. So they maintain that mutation. Maybe it's good for the virus, or at least it's part of the virus. And then there was another random mutation here and not in the other sample. And so you can start to see how we can build this sort of web of how the genome is changing over time. And it's important to remember, though, that with real data, you often don't have access to every single person's sample and every single person's genome. And so in a real scenario, you might have something that looks like this, where you don't really know what the genome here looks like, but based on your knowledge of how mutations happen and what evolution looks like, you might be able to take these 2 samples and look at them and guess what this looks like, right? You might be able to guess that the person who infected these 2 probably had that gene mutation, right? Because the chance that this mutation happening randomly in both of those people is kind of low. It's actually very low. And so that principle is what a lot of building a phylogeny is based around. There's a lot of algorithms and software that are entirely based around how do we, based on just these 3 pieces of data, build a tree. And so now if we take the arrows and just make them a little bit more square than they are right now, we can easily see how this turns into the phylogenetic tree. And so we use this software to sort of infer this phylogenetic tree. And each time, the pink tips here are the ones that we actually have data for, and the black ones are the one where we had to infer it based on the data that we have. And just one final step to make this look more like the tree of life I showed you before is we can literally just rotate this tree. My field tends to show them in this orientation, but they're both trees. And one other important thing to note is that the x-axis in the tree represents, at least in this kind of tree, the number of mutations that are occurring. So you'll see that this line is a little bit longer than this line because this one has an extra mutation. So that's just how basically to read a phylogenetic tree. And I promise this will come in handy, so I'm going to show you some mumps trees later. But before we get there, let's just talk a little bit more concretely of what we can actually learn about an outbreak from this representation. And so let's say we have a slightly bigger, more informative tree. So I basically just added some data to this. And I've labeled the samples or the people from which each of the virus comes from. And based on our new knowledge of phylogenetic trees, we can look at this one and draw some conclusions pretty quickly. So the first one you can draw is maybe that sample 2 and 3 are more closely related to each other than they are, for example, to sample 4. And sample 4 and 5 are more closely related to each other than they are to the other samples. And that actually really, that fact alone allows you to answer some questions about what's going on. So one example that I can give you is, let's say that samples 2 and 3 are from people who got infected in the United States. And samples 4 and 5 are from people who got infected in Canada. And now you have a new person come into the clinic, and you're not able to do contact tracing on them for whatever reason. They have no idea who they got infected from, where they got infected. And so when you can't answer that question by asking them, we can use this phylogenetic tree to give us the answer. So for example, if sample 6 were to come into the clinic, and we're able to sequence that person's virus, we can know that, oh, okay, maybe they have a virus, or we know that they have a virus that's more similar to what we saw in Canada than to what we saw in the United States. And therefore, we might be able to infer that that person got sick in Canada. And now we know something about the spread of this virus. So I'm going to talk very soon about how we did this with mumps virus, but first we'll just take a short break for questions. Yeah.
Isn't it like the longer the virus is around, the more likely it's to have like a weird mutation? That's not like a mutation that's more effective, or like worse for you, or something like that.
Yeah, the question is, if a virus is around for longer, is there more likelihood to have a mutation that like affects the virus? Yeah, that's a great question. So the rate of the mutations happening is actually pretty constant. So a virus sort of replicates at a certain rate and the mutations happen. But it does sort of make logical sense that if you have a virus around, like in the human population for a really long time, you just have more mutations. And if you have more mutations and the chance of each of them having an effect is somewhat equally likely, then yeah, if you have more mutations, you might get one that helps the virus. And so that's why it's often really interesting to study these viruses that are spending time in human populations in outbreaks to sort of see what happens and to try to maybe even predict what might happen next.
You said that mumps are. So how exactly did mumps?
Yeah, so the question is, if mumps is only transmitted by humans, how did mumps come? That is a great question. Yeah, I mean, I think it's a great question because there are some viruses where we know, oh yeah, the virus used to be in this other animal and now it came into humans. We don't know that specifically for mumps. It probably has to do with sort of how mumps as a species evolved. There are lots of other viruses that are somewhat similar to mumps. So my guess is that at some point, all of these other viruses, like one of them mutated enough to create what we call mumps now. But I don't know really like where the virus came.
So in certain cases of transmission, say for example, Ebola, running through the population, susceptible, something else, could it just run out of people?
Yeah, so the question is whether a virus at some point can basically infect everybody who can get infected and then run out of people. And that's actually something we definitely see with viruses like Ebola. For the big outbreak that we've all heard of, there were lots of small outbreaks in villages where the virus basically just infected, killed everybody in the village, and then it stopped, and that's why we weren't seeing bigger outbreaks. With something like mumps, that's definitely possible. It's also in the interest of the virus for that not to happen. So sometimes you get mutations that actually slow down its transmission so that it doesn't sort of run out of places to go. So that's something that you actually see if you see a virus in a human population for a long time. You often see it become less infectious so that that doesn't happen. But if it does happen, it can. Keep going.
Keep going.
Okay, keep going. We're gonna get a snack break later, but we'll go a little further first.
Yeah.
All right, so now we're gonna apply these ideas of phylogeny to the mumps virus outbreak. And specifically, we're gonna try to answer one of the questions that I think was on everybody's mind at the beginning of the outbreak, which was, how did mumps get here? Why is it here? Why is it in Massachusetts? Why is it at Harvard? And because we hadn't seen a lot of mumps in the United States before this outbreak, and because there's a lot of mumps in other countries, one of the guesses was, oh, maybe somebody from Massachusetts went to another country, got infected there, and brought back the infection. And as you might be able to guess, this kind of analysis is very useful because we're able to match up the virus to where in the world looks like that virus. So I'm going to show you a much bigger tree than this. This is a phylogenetic tree with way more tips. I'm going to zoom in a moment, so if you can't read it, that's okay. But basically what you're seeing is hundreds of tips representing hundreds of samples. And this is actually the work that I've been doing for the last couple of years. It's been a really big collaboration with my lab at Harvard and the Broad Institute and the Massachusetts Department of Public Health and Harvard University Health Services and the nationwide CDC to collect these samples, analyze them, and try to figure out what's going on. So what I'm showing you is, in this case, instead of coloring all the virus tips pink, I've actually colored the sequence of each virus by the state or the region in the United States from which the sample comes from, so that we can do exactly the sort of analysis that we saw in the previous slide. So what you're seeing is, if you can read this, is that there's a lot of blue on this tree. And blue represents Massachusetts. And the reason there's so much blue is because we were primarily working with the Massachusetts Department of Health, and that's where we got a lot of our samples. But we also got samples from around the country, from the CDC. And so we're going to zoom in on this tree a little bit. Now I've just sort of blown up this top region because it illustrates the point I want to make. And what I think the first thing that you see in this zoom in is that the blue tips are sort of interspersed with all of the other colors. And so what we're not seeing is what I showed in the example. We're not seeing that all the samples from Massachusetts look like this and all the samples from somewhere else look like that. And that's what you would expect if one person brought the virus into Massachusetts and that's what started the outbreak. So instead what we're seeing is that a very similar version of the virus is actually in lots of different places in the United States at the same time. And so what that suggests to us is that this outbreak or the outbreaks in the United States weren't caused by individual introductions from elsewhere, but actually represent a mumps virus that's moving all around the United States, right? Because if you have transmission in all over the country, then you're more likely to see very similar versions of the virus all over the country, rather than sort of different versions that come in from other places. And so this is basically suggesting to us that there's more mumps in the United States than we thought there was, because we're seeing a lot of evidence for it transmission across the whole country. And so just to sort of visually show what I'm saying is, instead of having isolated outbreaks into various states, we're seeing something that looks a little bit more like this. And what we think is that the reason that we're getting these big outbreaks, like the big outbreak at Harvard and the big outbreak in the Midwest in 2006, is not because there's like a new mumps strain coming in, but actually just because mumps virus is being transmitted everywhere, and then every once in a while it gets into one of these close-knit communities. Remember, I was telling you that mumps is often transmitted in places where people live really close together, because that's how it can infect lots of people at once. So every once in a while, somebody who's in college gets mumps, they bring it back to their university, and because they live closely with 400 other people, suddenly we get these really big mumps outbreaks, not because we're getting a new strain of mumps. So this definitely changes how we think about mumps in the United States, and is also a little bit hard to reconcile with this fact that over 90% of US children have at least one dose of the MMR mumps vaccine. So if we're all vaccinated, why are all these people getting mumps? And that was another central question of this study, and the next thing I'm going to talk about. So before we get into the answer to that question, why all these vaccinated people are getting mumps, let's just talk a little bit about vaccination, what happens in your body when you get a vaccine. When you get a vaccine, you're injected with this mystery fluid, but it's not actually mystery fluid. You're often actually injected with the virus or with the pathogen that you're interested in fighting off. And the reason that you don't get sick with that virus is because the vaccine often contains a variation of the virus that doesn't make you really sick. So it's often grown in the lab and changed a little bit so that you won't actually get the disease. Or sometimes you're injected with just little parts of the virus and not the whole virus. And the reason that it's important that you get injected with the actual thing you're trying to fight against is because in response to a virus, for example, the way your body fights that off is by creating these special molecules called antibodies. And the antibodies that your body creates are really, really specific. And each time you're infected with a different pathogen or virus, you make a different antibody. So here I'm just showing 2 examples of what a virus particle might look like, and showing you that in each case, you're going to get an antibody that looks a little bit different. And that's because each virus has sort of a different looking outer coating, and the antibody is made so that it recognizes only this outer coating. So this antibody will not recognize this shape, and this antibody will not recognize that shape. And once you have an antibody, the next time you're exposed to that virus, your body already has those ready. It's really easily able to recognize that you have something bad in your body, and it tells other cells in your body to go kill that virus. And so the reason we vaccinate is because we can sort of prepare for the inevitable exposure of this disease. We can make these antibodies and have them ready to go. If you don't have the antibodies already, that's when you get sick. When your body is trying to fight off the disease and make the antibodies at the same time, that's when you get sick. But of course, we've been talking about this idea of genomics and mutations and DNA, and sometimes you get mutations that can actually affect the binding antibodies. So here I'm showing you again an example of what the virus DNA sequence might look like. And this sequence will make a virus that looks something like this. And something that I'm not going to go into very much today, but I'll just sort of mention briefly, is that different parts of the virus DNA controls different parts of the. So if you're familiar with the idea of genes, so each gene has a slightly different function, has a different role in what the virus does and looks like. And so in this particular example, let's say that this left part of the virus DNA controls what this outer molecule looks like. And that's important to recognize because if there's a mutation in that specific part of the virus DNA, that can maybe change how the antibody binds. So for example, if there's this mutation in that region, these circular outer parts, this is obviously just a graphical representation, but might change to rectangles. And if you have rectangles on the outside of the virus, then these special antibodies are made that are really specific to that particular virus aren't going to be able to bind anymore. And this process is why, for example, you need to get a new flu vaccine every year because the virus is changing so much that your body, even though it makes antibodies and it protects you from one season, the virus changes and now your antibodies don't recognize the new virus anymore. And this is different for every virus. Some viruses don't change that much. For example, you probably got the polio vaccine as a kid and that's going to protect you for your whole life. There's no reason to believe that polio is going to change. So that's just one example. So we're going to take one more quick question break, and then after that, we'll talk about how we can find out if this is happening in mumps or not. No questions. Well, maybe this is a good time to take the longer break, and we'll find out what's going on with mumps and whether you're protected from your vaccine.
All right, so we'll take a few minutes, go get some more snacks. This is the last lecture, so we have to eat them all. And if you have any questions that you think of, you can come ask Shirley, and we'll come back in 5-ish minutes. All right. Cool.
Yes, animals is like an RNA virus, but it's not. It's not how we would explain RNA viruses. DNA is not. You can. I've done that before. Yeah, so yeah, mumps is an RNA virus, as is most of the. You can't own it with PowerPoint. She is looking at PowerPoint. Most of them sound like maybe she's using. Yeah, the ones that make favorite is actually using.
Makes sense, yeah, I guess that. So
Yeah, okay. You probably do. How are you actually doing it? So like what if instead of clicking that full screen, let's see.
Yeah, but things like that.
If you go to slideshow.
I think.
Okay, so because I know the people in my lab use PDFs. They don't use slides. They make all their slides in LaTeX and then like export to a PDF and then so okay.
Yeah, it's really just like so.
You gonna give us some feedback? Because I like this.
It's really nice.
Yeah, and now is the bottom half is the stuff that's useful. So if you really like to get that, yeah, we'll figure it out. We'll figure it out later.
Although it's good.
Figure perforated.
Yeah. This is cool. It's interesting because a lot of my stuff is basically making clustering of different stimulus representations of brain. Oh, hey. I've done these kinds of things. Or what distance makes up there?
Super exciting. There's some of the underlying zero questions. It's like really
It's really clear, yeah.
Yeah.
I'll also get more. Well, actually. Yeah, I mean, it's how big.
Yeah.
This is my precious baby.
That's really cool. So we do it. Just for suggesting. That's right.
Yeah. Yeah. Our office is like, if there wasn't a wall between us, there would be like. That's why I get daily day content.
Like all the viruses, cells, and also.
Well, so the little virus part of the cells, I gave the vaccines all like 3 years ago,
Okay.
and Joy grew those.
Joy, okay, I was saying, I know that I've seen other people use them.
She's incredible.
She's still around, or she graduated from
She graduated last year.
Okay, I'm not sure.
She's only 1 year above me. Yeah, so she.
Should bring her back. So good. Yeah. Except where I use a lot of it. It's like restrictions in her complex. So good. It's like water. I like it.
I like tried before.
And PowerPoint shapes. Not good. Yeah. Yeah.
You can say your question.
Save my question. All right, I have 2 questions.
Good. I have questions too, but I feel like they're too specific.
You're allowed to ask questions. Ugh. Gosh, I'm dropping kale all the way. I feel like I'm following Katie's shoes. She always brings her dinner to the lectures, but her dinner always includes a whole purple sweet potato. Like, and she just sits there and just eats a purple potato like it's an apple. I haven't quite made it to that point yet. I don't know. I never remembered actually looking at her face. I just told the whole internet about Katie's potato. Great. The whole internet.
2 minutes. 1 minute.
Okay. 59. 58.
Do the slides correctly this time.
No, but we figured out a different way that you
There's
can
a
use.
slideshow mode.
Yeah, it's different. I didn't realize slideshow was different than full screen. That's the kicker. All right, I'll go turn on the camera again.
Did she turn her mic off?
Oh yeah, good call. So now-
All right, cool.
Yeah, should we finish it out? Does anyone think of any questions while you were getting some snacks? I actually did. So this is kind of main questions about mumps. Is the main kind of risk, or the reason why it was so bad, just that it has the tendency to increase people's risk of getting meningitis and these other encephalitis, or can mumps itself kind of be dangerous or fatal or something?
Yeah, so the question is, yes, whether mumps is all that bad, but like if it's ever fatal or whether it's just these reduces your risk for other things. Yeah, it's definitely an interesting disease to study. Like, I'm coming off of studying Ebola and Zika, and like those are clearly like, oh my god, they cause really bad things. Like, everybody should worry about it. Mumps is almost never fatal. The real risk is the complications. I think the biggest risk is that the chance of infertility in men is actually rather high. So that's one thing. It used to also be one of the leading causes of deafness in children.
Yeah, I study hearing, so I knew that it had a connection with deafness.
Yeah, so
Not
it's
so much
not
anymore.
something that's going to kill you, but I think because it was so widespread and because it had the chance of serious complications, it was like an early candidate for vaccination. And then I think nowadays it's actually both important to realize why we're all getting mumps, but also a really good, and I'm going to talk about this at the end, but a really good example of like the potential of this kind of research, so that when something like Ebola comes back, we're like ready to do it and we have all of the technology.
So what's the mechanism for infertility?
It has to do with the fact that
Repeat the question.
oh, sorry, thank you. The question is what the mechanism is for infertility. And so the thought is that I don't know the like really details of it, but just like it causes swelling in a gland in your cheek, it sometimes causes swelling of the testes, and something about that process that might be known, but I'm not exactly sure, because sometimes it did. But that's why it's just in men. All right, well, if there are no more burning questions, we'll talk about whether this process of mutations that might affect antibody binding is actually what's going on in mumps. Because if this is what's going on, it's really easy to explain why people who are vaccinated, like Harvard students, students that in universities are some of the highest vaccinated populations in the country, because you often have to get vaccinated to go to school. So it would be really easy to explain, oh, there's a mutation in this region, it makes the antibodies not bind, we just need to update the vaccine and we'll be able to stop this. So we looked at all of the genomes that we had, hundreds of genomes from Massachusetts, from around the country, and we didn't actually find any mutations in this region that were reasonable to explain why people were getting sick. So there weren't really any permanent mutations in this region that we think can affect antibody binding. We did see lots of mutations in other regions, but they shouldn't actually affect this particular process. So they shouldn't actually tell us anything about whether antibodies can still bind. But what we think is actually happening is, even though the antibodies can still bind, we're finding, or that we think that people have fewer amounts of antibodies in their bodies over time. So this is a process called waning immunity. And it's not unique to mumps. We've seen it in other cases before, where that's why sometimes you need to get a second dose or a booster of a vaccine, because over time, for some reason that we don't fully understand, your body just stops making as many antibodies. And so even though your body has some, if it doesn't have as many, you're not really able to fight off the disease and you can still get sick. And so this is what we think is happening in mumps. And some other research that other labs have been involved in have estimated that when you get vaccinated for mumps, you're fully protected for maybe 15 to 20 years, but after that period, you're able to be infected again. And that lines up really nicely with the fact that we're seeing lots of college students getting sick, because the mumps doses are administered to toddlers. So you're a toddler, you get your second dose when you're a few years old, and then 15 years later, you're a college student, you're in this close environment, and suddenly the chance of getting mumps is higher. And so I think this begs the question of, then why are we seeing the mumps outbreaks now? The vaccine has been around since the 60s. It's been around for decades. Like, why is this phenomenon happening right now? And I think that the answer to that question comes from this idea that even though we think that your immunity wanes over time if you were vaccinated, we think that actually getting mumps protects you forever, for your lifetime. And this is actually a really important nuance, because if you think about who is able to get infected in the population. So 60 years ago, everybody had mumps, everybody was immune for life. So that group of people has been getting older and older. And for a while, all the kids are protected. And then as time goes on, those kids are no longer protected, but all the adults are protected. So even though the adolescents can get mumps, there aren't a lot of adults in the population who can get mumps, so there's not a lot of avenues for the virus to be transmitted between. But now, if you fast forward another 30 years or so, the population of adults who are protected has gone way down because the group of people who are immune for life is just getting older and older and is a smaller and smaller percentage of the population. So in another 50 years, there won't be people in the United States who weren't vaccinated for mumps, and so there won't be that population of people who are infected. And so now, as time goes on, the population of adults who can be infected by mumps is getting bigger and bigger. And as there are more people who can be infected by mumps, just the chance of transmission is higher and higher. And actually, there's been some other groups, some of our collaborators and other labs that have done some modeling about what would you expect if we had this exact scenario happening, where the vaccine wanes after 15 to 20 years, and the vaccine was developed in the late 60s. And these models are actually really interesting in that they show exactly this, that about 60 years after the vaccine is developed, you see a spike in the number of mumps cases. So this sort of all lines up with this theory that maybe the vaccine just doesn't last very long. And this has actually led to a recent recommendation by the CDC. So last October, the CDC, which I realized I didn't define before, but for anybody that doesn't know, the CDC is the Centers for Disease Control and Prevention. It's a national government body that is in charge of hopefully preventing all of us in the United States from getting things. And they released a recommendation saying that people who are in high-risk situations in outbreaks should get a third dose of the mumps vaccine. And that's based on this idea that your body isn't making as many antibodies, so if you get another shot of the vaccine, your body will start making more of them. And so I think it remains to be seen whether they'll be recommending more vaccination over time, whether this will be effectively utilized in outbreaks. There wasn't a lot of vaccination in the most recent outbreak. So I think we'll see if that makes a difference in the number of people who can get mumps. But the good news is that the vaccine still works, and there's no evidence to show that it's not working. And one thing that we did notice in this outbreak is that the symptoms of people who got mumps were maybe milder than we might have seen, and very few people had the complications that we're worried about. So even though you're still able to get mumps, it might reduce the severity of your symptoms if you're vaccinated. So vaccinated for mumps and everything else. All right, so the last part of my talk, I just want to talk about the future of outbreak response and sort of get to Dana's question about, okay, but this is just mumps. Nobody's dying from mumps. Like, how can this really be extended? How can these lessons be taken forward to scenarios where we really need to contain the outbreak? And I'm going to talk about this primarily through actually an example of mumps, because I think it really illustrates why we want to do this research. So yeah, we figured out the vaccine still works, but sort of what is the potential of really being able to track who's infecting whom and how the virus is spread? So I'm going to talk a little bit about a really specific example of mumps here in Massachusetts. And what I'm going to tell you about is there were actually 2 waves of cases of mumps in Massachusetts in the 2016-2017 time period. So the first wave of cases was in 2016, and the number of cases in any given week is shown in orange. And after that, there was the wave of cases in 2017, and those are shown in red. And what's interesting is there's actually a short number of weeks between those 2 waves of cases. And additionally, the cases were in 2 very different populations. In 2016, most of the cases were in college students at Harvard and elsewhere. But in 2017, most of the cases were actually in a community in the greater Boston, Massachusetts area. I can't tell you exactly where there weren't really connections to college students. It was a generally older population that wasn't enrolled in universities at the time. So there was no apparent connection between what was going on in college campuses and what was going on in this community. And so based on the fact that the two outbreaks were separated in time and they were in different groups of people, it would have been a pretty logical conclusion to say, maybe these are totally separate outbreaks. Maybe they're caused by different introduction, right? Maybe the one in college student is the one that's circulating everywhere, and maybe the one in the community came from a specific place, and they're different types of virus. But one thing that we can use the genomic data for, the DNA data for, is to actually try to get an answer for this question. And so here, we're going back to this idea of the phylogenetic tree. I'm showing exactly the same phylogenetic tree, but this time I've zoomed in to the bottom of the tree. And the reason I've done that is because all of the samples from this community are clustered at the bottom of the tree. So all of these samples in blue from Massachusetts are really similar to each other and are all from patients who are affiliated with that community. But right near them on the tree are all these samples that are affiliated with Harvard, with students at Harvard University. And what you can see from this is that even though they're slightly different clusters, they're actually really related to each other. They're much closer to each other than, for example, some of the other clusters in this same outbreak. And so what that suggests is that these aren't separate introductions, these aren't totally different outbreaks, but they're actually related. And if we think back to what we learned about phylogeny earlier, we can actually conclude that it looks like this community outbreak seems to have come from the Harvard outbreak. So it seems like the virus evolved forward in time, and that we're seeing sort of what we call a spillover event, spillover from the Harvard community into the larger. And so while this was something that was like abundantly clear from the genomic data, there were really great records and really great contact tracing at Harvard, the Department of Public Health didn't immediately pick up until we looked at this. But of course, once we were able to identify this connection, we went back to the contact tracing data called up Harvard University Health Services, and we sort of put all the pieces together and realized that there are actually 3 individuals who are affiliated with both the community outbreak and the Harvard outbreak. So we were actually able to put all of these different pieces together and understand that there was a link that we could find. And I think this is a really nice, beautiful illustration of how the contact tracing data and the genomic data can sort of work together to give you a really complete picture of what's going on. Because if you had done the contact tracing, even though that data was there, you might have not known to look for it, or it might have been, in the case of the Department of Health, they had so much data that they were collecting from different people that it just wasn't apparently obvious. When we added the genomic data, we really saw this link that we were able to confirm. And just to take this sort of one step further, as I mentioned, there was contact tracing that went on. In particular, Harvard did a really excellent job of contact tracing. There's lots of really detailed records of all the students who got infected. But even though they did a really incredible job of putting all this information together, if we look at sort of the known links between patients at Harvard, it's actually a pretty sparse picture of what's going on. And that's because, as I mentioned, sometimes you don't know who infected you. You know, you go to a large gathering and you interact with 50 people, and that was 2 weeks ago. You don't really know which of those 50 people gave you mumps. So there are very few known connections between. So on this plot on the left, I'm showing each circle is representative of a virus from one patient, and the color represents the school where they're from. And the red lines represent these known links. So places where contact tracing told us definitively these 2 people are connected in transmission in some way. But if we use the genomic data, we can actually get a much more complete picture of what's going on, because we have this information about the virus samples that doesn't rely on us knowing exactly who infected who. We can actually infer that from those relationships between the DNA. So here on the right, I'm showing a reconstruction of what transmission might have looked like based on just the genetic data. And the shading of the lines has to do with the probability that those 2 people actually infected each other. And so I think, again, this shows sort of what genomic data can tell you that this contact tracing data might miss. And not because we're not trying hard enough, but just because it's really hard to trace transmission with some of these outbreaks. But I think one of the coolest parts of this project is when you put those two types of data together and you get really the most complete picture of what's going on. You're able to see the relationships between the DNA, but also look at the relationship, for example, between different schools. So I told you that each dot, the color represents the college or university from where that person is affiliated. So now you can start seeing trends about, there's a lot of connections between this blue school and this green school. Maybe there are links, maybe there are transmission chains between them. And you can also start to sort of derive actionable conclusions that a university might be able to use. You might be able to identify people like this who infected lots of other people and trying to figure out, how can we change our containment strategies to target those people. You might see that certain groups of people have lots of different links between them. They're really tightly connected. Maybe they're involved in a certain type of activity and we want to make sure that that sort of group of people gets tested for mumps slightly more often or something like that, just trying to think about how this is all done. And so I think that this is sort of a retrospective analysis of what happened in mumps last year and 2 years ago. But nowadays, we've developed the technology where you can actually do this pretty close to real time. And that was a big barrier for a long time, the ability to sequence samples fast enough to learn from them. But now we have the ability to sequence virus samples really quickly, in a matter of days or weeks. And I think most importantly, something that we were able to do through this project is develop the collaborations between the different players, the different institutions, the different people who need to work together to sort of this outbreak. I mentioned that this whole project was a big collaboration between my lab, the Department of Public Health, the CDC, Harvard, all of these pieces that really need to fit together to understand and try to fight an outbreak. So I think that mumps, not only do the data illustrate the power of doing this genomic analysis, but have also sort of helped us build the foundations that we need to hopefully address any future outbreaks that come our way at Harvard or elsewhere. So just to wrap up, I just want to mention that now that we're building the technology to do the sequencing really fast, we're applying these techniques not just to mumps, but also to the scary viruses. We've done a lot of this analysis in Ebola, we've done it in Zika, and we're getting better and better at making this data available, analyzing it quickly, getting the genomes really fast. And so I think, before I thank the news, I owe a really big thank you to all of the people who make this kind of work possible. This is not the kind of work that one lab can do by itself. These are big collaborations, lots of help from lots of people. And I thank you guys all for coming and for hopefully learning a little bit about mumps.
Questions? I have a question. Yeah. Can you speculate on if you think a third dose will be routinely implemented during childhood, or if they want to use it more during these outbreak scenarios?
The question was whether I could speculate on whether the third dose will happen in childhood or in outbreak scenario. I'm not sure I'm exactly the right person to speculate on this, but I think the implementation of a third dose is going to be really a cost-benefit analysis that's done with what is the cost of sort of nationwide implementation of additional vaccines versus the cost of just doing it in sort of a reactive setting. And I think that there's going to have to be more studies, both in actual outbreaks and modeling studies, to show how effective can we be if we just do what we call reactive vaccination, which is just vaccinating in outbreaks. And I don't think that those studies have really been done, or not to the extent that we want. So we'll see.
Is there something special about mumps virus that causes it to be more susceptible, or I guess us to be more susceptible to this waning effect?
Yeah, so the question was, is there something special about mumps that makes the waning immunity happen that doesn't happen? I don't think we actually know. It's something that we've seen in some viruses and not others, but my understanding is that it's not a well-characterized mechanism. mechanism. It's actually pretty surprising and impressive that we don't know a lot about how viruses make us sick and all these things that I think are pretty related to how our body's immune system responds to them. So yeah, not just mumps, but we don't really know why. Like especially since we get mumps and measles together, and measles doesn't seem to have this problem. Yeah.
Do you have any statistics on what the rate of these complications is? How serious a problem this is?
I did. I'm not sure I can recall them off the top of my head. I want to say that something like 1% of males infected with mumps, somewhere around there. I don't know if you have a better answer.
It's like less than.
Yeah, so it's not a huge number, but just when you were getting hundreds of thousands of people, and the fact that it was affecting children, I think made it just a candidate for vaccination.
So when you're talking about using these sort of phylogenetic trees and modeling for future outbreaks of these more deadly diseases, what sort of factors make it so that you can't just take the strategies and models and things you have and just apply it to another disease? What makes these other ones potentially more complicated or difficult?
Yeah. So the question is, what might make this challenging to apply like phylogenetic trees and things like that to other outbreaks and viruses? I have two answers to that. One is something that I've learned working on mumps and also different viruses is that viruses are very different from each other. Like lump them all together and we're like, those are the viruses, but there's so much diversity within viruses, and each virus mutates at a different rate and has different patterns and has different times to infection. And I think each time we do this analysis on a new virus, we sort of have to re-explore that space. How fast does this one mutate? Are we even going to see mutations? If we don't see mutations, how do we deal with that? So I think that's one really big part. I mean, I think another part of that is some viruses are actually harder to sequence than others. So when we were studying Zika, that virus is just, for a variety of reasons, way harder to get a sequence out of. And so that's like another hurdle. But I think the other answer to that question has to do with sort of the building collaboration side of things. Right now, a lot of these collaborations develop as a response to outbreaks. And so things like getting permissions to do things takes a really long time. Also, the worse the effect a virus has on a human and the more like stigma there is, the harder it is to kind of get the information we need to study it. So I think those are sort of the two types of barriers. So it's definitely being done, but something that's still building and we're still learning how to deal with.
There's been a lot of talk of vaccine deniers and people who are turning against vaccines. Has that had anything to do with this mumps outbreak, or is this really just about the waning immunity component?
Yeah, that's a great question. So the question is whether the sort of anti-vaxxer rise has anything to do with why we're seeing more mumps. I think the answer is probably not. I think this outbreak sort of happened too soon after we were seeing rates of non-vaccination increase. Like, I don't think we'll be seeing effect quite so soon. Not to say you shouldn't get vaccinated, but I don't think there's a direct correlation for this particular outbreak. I think it could sort of exacerbate the effects as if people are not getting vaccinated, the rate at which that population increases of people who are susceptible could increase much more rapidly. And we might start seeing outbreaks in non-college ages. We might start seeing outbreaks in kids again, which we're not seeing. But I don't think that had a direct effect on this. But I think there are other diseases for which we're starting to see effects. Like we're seeing some measles outbreaks that might have something to do with that.
All right. Well, thanks for coming out, you guys. And if you haven't had a chance, we would love it if you give us some feedback on our audience survey so that we can learn how we can do better, and some feedback for our lovely speaker. And we're off but we'll be having another lecture series in the fall, this time on the Boston Medical School campus. So sign up for our mailing list, follow us on Facebook, all that kind of stuff, and you'll hear about those lectures. And there's a question on the survey asking what kinds of topics you want, and we actually read those and try to find people with those topics. So if there's something you want to hear about, please write it down. Thanks.
Awesome.
But it's more. Oh, yeah.